Welcome to Biology Made Easy. We are starting our lessons in genetics today. And in this lesson, we are going to explain various genetic terms. Welcome. Genetics is a science that deals with heredity and variation. Well, heredity is transfer of characteristics. I have this parent, male, female, they produce these offspring. So transfer of the characteristics of these two to their offspring, that is heredity. And then also genetics is a science that deals with variation, and variation is differences among the individuals. So you have differences, you have these two with these children, all of them different in some aspects. That is variation. Now, genes determine characteristics. So this male parent, this female parent, they produce these offsprings, transferring their characteristics in the form of genes, because genes code for protein, and protein determines the characteristics of individuals. So terms we will meet include genotype. Genotype is the genes in the organisms. Phenotype is the outward appearance. Well, these organisms transfer their genes. You can't see the genes, but you can see the effect of the genes because of the outward appearance. And the outward appearance is the phenotype. The outward appearance can be observed, it can be measured. Genotype, genes cannot be seen or measured, but it's measured in the form of the phenotype. Then you know chromosomes bear genes, and we have this chromosome and that chromosome. In diploid organisms, that is organisms that have two sets of all chromosomes, you have one set of the chromosomes from father, paternal, another set from mother, maternal. Chromosome bear genes, a number of genes. So I have this chromosome has gene A, another chromosome, gene small a. Genes and alleles are represented by letters in genetics. Alleles are different forms of the same gene. One gene can have many alleles, many different forms. For instance, a gene like gene for flower color in a plant, the gene for flower color can have many different forms. That is alleles. The gene can be a gene that produces red color, that's an allele. You can have another allele that's a different form of the same gene that produces white color. You can have another allele that produces yellow color. In diploid organisms, that's organisms that have two sets of chromosomes, any organism can have any of these two. So you can have some organisms that have two of this, big A, big A. You can have another organism that have two of this, small a, small a. You can have another organism that have two of this A, Y, A, Y. There's also the term homozygotes. Homozygote is a cell or organism that contains two identical alleles for a trait or character. With these alleles, these organisms are homozygotes. Each chromosome has the same allele. So we have big A, this is a homozygote. Small a, this cell or organism is also a homozygote. A, Y here, this also homozygote. Then you have heterozygotes. Heterozygotes organisms have two different alleles for their character. So I have big A, heterozygous, big A, Y here, heterozygous, small a, Y here, heterozygous. So all these are heterozygote. Good. Then we will look at the term dominant allele. A dominant allele is the allele that shows its characteristic in the phenotype, that's the outward appearance of the organism, in such a way that the expression of the other allele at the same locus of the homologous chromosome called recessive is masked, does not show up. So we have these alleles. If this is a dominant allele in this heterozygote, this allele will show its characteristics over and against this allele. So this individual will be red. The red allele is dominant, meaning it shows its characteristics in the outward appearance of the organism if masking the appearance of the other allele. So this allele is a recessive allele. So this one becomes a recessive allele because even though it is present, it doesn't show up. And if big A is dominant, then this one too is recessive. And so this organism here will be red. Now, the recessive allele shows its characteristics in the phenotype. This will be white. The recessive alleles show their characteristics in the phenotype when they are double. The alleles are double. So this one will show its characteristics as yellow. Now you can have dominance in humans. A number of traits that show dominance. A typical one is tongue rolling. Tongue rolling gene is big T, is dominant over non-tongue rolling, which is small t. So you see this big T tongue rollers, big T, small t tongue rollers, and these are non-tongue rollers. The recessive alleles always show themselves when they are in the homozygote state. Now we come to another term, codominance. In codominance, both genes contribute equally in the phenotype, contributes equally in showing the phenotype. So big A is a gene for red, small a is a gene or allele for white, but then when there's codominance, the flower looks pink. Both alleles contribute equally in the phenotype. Example in humans is blood group A and blood group B are codominant. We'll come to blood groups in good time. Then you have another term to explain partial or incomplete dominance. In this case, if you are still using the flower colors, big A and small a, if partial or incomplete dominance will not be pink as when there's codominant. But then one of the alleles will contribute more than the other. And so the flower color can be light red or white with some tint of red or patches of red. And in humans, example is the sickle cell anemia trait. Now, another term, hybrid. A hybrid is an offspring produced from parents that are genetically different. And so big A, that is red flower, white flower, and they cross, you have an offspring which is big A, small a, and it's a hybrid. So hybrids are heterozygotes, and the heterozygote was produced from pure breed. Pure breed, pure stock, true breed means the organism is homozygous for all its traits. So if you have an organism that is big A, it's a pure breed. Small a, is also a pure breed, homozygous organism or traits. Now, breeding true is when an organism, e.g. a plant, is celled, that is self-pollinated, generation after generation, and the same characteristics is seen in the offspring, then the organism is breeding true. It means it's a pure breed or a true stock. So if this plant is breeding true, it means this is the flower. If it's self-pollinated, pick pollen grains from the anther, put it on the stigma, collect the fruits, plant the seeds, get a plant that is exactly like it, red flowers, and this is done from generation to generation, and you get the same result of red flowers. Then this plant is breeding true. It means that red is dominant. So we have homozygous dominant red. So you have big A, and that is the only way that you can produce red flowers all the time. You know, you can have red flowers if it's a heterozygote, if gene red is dominant. But if it's breeding true, all the offsprings from generation to generation will be red. If it was a heterozygote, then at a point you will have some white flowers coming in. So you can have this homozygous recessive, which will also breed true. If it's self-pollinated from generation to generation, it will produce white flowers. That is breeding True. Now we want to look at generations, F1, F2. So if I have homozygous parental stock, this dominant, homozygous, recessive, homozygous, when I cross it, we'll have this heterozygotes. All of them will be heterozygotes. That is the F1. And the F2 is selfing the F1. So this F1, we self it. The offspring produced gives us the F2 generation. Epistasis is a condition where one gene suppresses the effect of another gene at a different locus. The suppressed gene is hypostatic, the suppressor gene is epistatic. Now let's look at the term epistasis or inhibiting gene. I have these two chromosomes, this and this, they are homologous chromosomes. I have this gene here, A, with its companion small a, they are producing an effect. I have this big B and this big B also producing an effect. This is on its locus. This gene is also on its locus. Now, if this gene, big at its locus, suppresses the effect of this gene at another locus, then this gene is epistatic. So this gene that is suppressing the effect is epistatic. This gene that its effect is being suppressed is hypostatic. Well, this is where we end today's lesson. Please give us your thumbs and kindly subscribe if the lessons have been found useful. Thank you very much. Goodbye.